Hello class, welcome to the module 2 of our molecular biology and diagnostics. For today's lecture, we will be dealing with DNA or your deoxyribonucleic acid. But before we head on to our main discussion, let us just have a quick review or recap of our discussion last meeting. On your module 1, we were able to discuss the introduction to molecular biology and its application in medicine. Next, a brief history of our scientists and their contribution in molecular biology. Next, nagkaroon lang tayo ng overview of our DNA. And lastly, your reading assignment, which is the central dogma of life. For your learning objectives, at the end of the session, the students must be able to describe the basic parts and the structure of the DNA. Next, describe the importance of DNA in molecular biology. Okay? Based on your reading assignment, what is the deemed importance of central dogma of life in our microbiology and in our life as well? Basically, the central dogma explains the flow of genetic information from your DNA to RNA to make a functional product, which is your protein. Again, the central dogma of life basically explains the flow of genetic information from the DNA to RNA to make a functional product, which is your protein. Sabi nga nila, kung wala kang DNA, wala kang protein. Because it aids in the synthesis of your protein, the DNA po. But how it works? Ano po ba yung process involved in the central dogma of life kung bakit ka nagkakaroon ng protein through your DNA? Paano po ba yun, ma'am? It starts in the synthesis of your DNA. Okay? First step, magre-replicate muna yung DNA mo. It will make a replica. Ibig sabihin, it will produce an identical DNA. Next, after replication, it is then followed by transcription, which is the conversion of DNA to mRNA. Okay? Transcription came from the root word transcribe, meaning to write something in a different way. Okay? Kasi po, it is called transcription because it deals with the same language, which is nucleic acid. Kasi nga naman si DNA, nucleic acid din siya. It is then transcribed to another language, which is nucleic acid pa din, nung na-convert mRNA siya. So, ang tawag sa kanya, transcription. Next, when mRNA is converted to, skip muna natin to, protein, in the process called translation. Okay, translation naman, it came from the root word translate, meaning to change to different language, which is protein. Kasi nga naman po, ang language natin dito, nucleic acid, nung na-convert na siya to another molecule, naging protein. So, ang term na natin doon, translation. Okay, balikan lang ulit natin, DNA. Nung na-convert siya to our mRNA or RNA, ang tawag doon, transcription lang. Na-transcribe siya to same language, basically parang nucleic acid. And then, nung na-convert naman yung mRNA mo to protein to another molecule, ang tawag naman doon, translation. Okay, ma'am, ano naman po itong reverse transcription na nasa harapan namin? Reverse transcription, tandaan natin na may mga living cells tayo such as yung mga virus natin, meron silang RNA sa kanilang cell. And then, the process of RNA, of converting RNA to DNA is called reverse transcription. Okay, basically, ito lang yung content ng ating central dogma. Nilagay lang natin siya sa tabular form. The DNA replication, ang template niya is DNA. Galing siya sa DNA. Ang product niya, since nag-replicate lang siya, DNA pa rin. Sa transcription naman natin, from DNA, na-transcribe siya to another nucleic acid, which is RNA. And the translation from mRNA, naging protein na siya. Na-convert na siya to another molecule, which is protein or amino acid. Take note that the product of transcription is RNA. mRNA is not the only RNA produced during transcription. You could also produce rRNA or tRNA, but the template for translation should be mRNA. Meron tayong emphasis dyan, pakitandaan po yan. Next, your deoxyribonucleic acid is the fundamental units of genetic information. It basically carries the hereditary material coming from our parents.
The deoxyribonucleic acid is the chemical basis of heredity. It is also the blueprint of living organism. And then usually, ang ating mga nuclear DNA, it is linear in form. Okay, kagaya nung nasa picture on your right side, pakitingnan, linear po siya. It is a double helical structure as proposed by Watson and Crick. Double helical, pansinin yung ribbon, double helix po siya. Okay, proposed by Watson and Crick noong 1953. Saan po ba nakalocate ang ating DNA? Okay, ang ating DNA, it is basically located in the cell, okay, living organism, particularly in the nucleus. Cell muna, tapos nucleus. Sa ating nucleus, nandun ang ating mga chromosomes. Ano po bang laman ng chromosome, ma'am? DNA lang po ba? Hindi po. Si chromosome po, it contains DNA and other proteins. May mga proteins pong naka-embed sa ating chromosome. Doon sa chromosome mo, may pagkahaba-haba tayong DNA. Kung pansinin nyo, linear in form, has double helix and base pairs. Pakitingin, base pairs po. Next, bakit po siya linear, ma'am? Pero coiled siya. Ano po ba yung responsible sa coiling ng DNA? Ito po yung histones. Okay? Importante yung histones kasi it is important because it is important in the packaging or the condensation of DNA. Okay? Ang ating histones ay basic in nature. Ang DNA naman natin, itong mga histones, ito yun okay? Acidic po sila in nature. So, nung nagsama yung basic at nagsama yung acidic, nagkaroon sila ng electrostatic attraction. Okay? That's what made the DNA supercoiled. Ma'am, bad thing po ba na coiled ang ating DNA? Hindi po. Okay? Actually, the supercoiling is needed to package the DNA inside the nucleus. Okay? Si histones, nakatulong pa siya kay DNA para mas maging coiled at mas maging packed siya, well and condensed para sa nucleus. Okay? Kapag ka na-embed na siya dun sa nucleus. Okay po ba? Next. Who discovered your DNA? DNA was first recognized and identified by Swiss biologist Johannes Friedrich Mischer. I don't know if I'm correct in pronouncing it right, sorry. In 1869, during his research on white blood cells, the double helix structure of a DNA molecule was later discovered through the experimental data by James Watson and Francis Crick. Okay, kung sino yung naka-discover and naka-identify ng DNA, si Mischer yun. Pero yung structure na double helical, through their experimental data, that was James Watson and Francis Crick. Do not forget, finally, it was proved that DNA is responsible for the storing of the genetic information of a human being. Did you know that most DNA is located in the cell nucleus where it is called nuclear DNA? Okay, kaya nga siya tinawag na nuclear DNA kasi nakalagay siya sa cell nucleus. So again, ang ating cell, meron siyang nucleus at nandun sa nucleus na yun ng chromosome. Sa chromosome, doon naka-embed ang mga DNA and other proteins. But a small amount of DNA can also be found in the mitochondria where it is called mitochondrial DNA or your mtDNA. So ma'am, ano po bang relevance niya and paano po nangyari yun? Okay, pakitandaan ng ating cell, meron siyang cytoplasm. Nandoon ang ating mitochondria. And then, yung mitochondria mo, meron pong mga DNA yan. It came from our mother, sa mother ng mother natin, mother of mother of mother of mother. In short, galing sa ating maternal line. So, how did that happen? During reproduction, yung sperm cell na galing sa iyong father, and then the egg cell that came from your mother. So, basically, ang egg cell is a complete cell, meaning may mga nucleus siya, may mga organelle, may cytoplasm, and may mitochondria. Okay, si sperm cell kasi wala namang mitochondria yan. So, nung nakabuo na ng zygote, doon po nanggaling ang ating mitochondrial DNA. But the mitochondrial DNA is unlike your nuclear DNA which is linear in shape. Ito, small siya and pabilog. Pabilog ang kanyang shape. So, wala siyang relation doon sa ating nuclear DNA when it comes to having the genetic information or carrying the genetic information. The
relevance or the significance of your mitochondrial DNA, its job is to aid in the production of ATP in the mitochondria. Okay, wala siyang kinalaman dun sa genetic information na located sa ating nucleus, yung mga DNA dun. Next, also your DNA is a great at storing data. 1 gram of your DNA is equivalent to 700 terabytes of data. And also your DNA from a single cell is 6 feet long if fully stretched out. So imagine niyo yung single cell pa lang, pag in-stretch out mo ng nakalinear ang ating DNA, 6 feet long na siya. Eh, di ba sa katawan natin sobrang daming cells, may mga trillions and billions of cells. So imagine gaano kahaba yon if fully stretch ang ating mga DNA. So, kumuha ako ng mga pictures sa Google to give us a better view of DNA and have a more simplified representation. So, di ba, ang ating DNA, naka-helix form siya and coil, di ba? Kung pansin nyo, naka-parallel. Imagine na lang natin na itong picture na to ay ang ating DNA that is parallel to one another to give us a better representation of the chemical structure. So, para mas maapag-aralan natin na maigi yung chemical structure ng ating DNA, humanap ako ng mga picture na nakalinear at mas simplified. So the chemical structure of DNA, DNA has 3 types of chemical component. Una na doon yung phosphate group natin. Okay, na-describe na natin sa ating last module ang content ng ating DNA, the chemical structure. Meron tayong phosphate, sugar, and then may adenine. Ang phosphate, it is located on the outside of the double helix. Do not forget, located outside of the double helix, 2 hydroxyl groups of each phosphate are involved in the phosphodiester bonds. And then the third is free and dissociate its proton at physiologic pH, giving the molecule a negative charge. Okay, do not forget that the phosphate group is the one that is responsible in giving the DNA the negative charge. So kapag nilagay natin siya sa isang electrophoresis gel, it will migrate towards the anode kasi negatively charged ang ating DNA. Next component is your pentose sugar, ribose or deoxyribose in a furanose ring form. Pentose kasi 5 yung kanyang structure, o, di ba? 1, 2, 3, 4, 5. DNA contains slightly different sugar because it is a type of ribose. Yes, it is a type of ribose, but it lacks 1 oxygen atom. Pansinin po sa picture sa baba, ang comparison ng ribose and deoxyribose. Dito kulang ka ng isang oxygen atom. Kaya siya deoxyribose. Okay, yung ribose mo, kompleto, merong oxygen atom. So, pansinin natin sa picture natin dito, yung phosphate group mo, nandito sa taas, yung inyong nitrogenous base na sa baba. Ano yung nag-hold sa kanilang dalawa? Si sugar. Sugar holds the base on one side and the phosphate on the other side. Thus, the sugar hold the components of the nucleotide together. Next is your 4 nitrogenous bases, naming your adenine, guanine, cytosine, and thymine. Do not forget that the purines, naming your guanine and adenine, has double rings. Okay, imagine natin marunong akong mag-drawing. Double ring siya. Purines, double rings. Pag pyrimidines naman or pyrimidines, single ring lang siya. Naming your cytosine and then your thymine. So, na-describe na natin yung tatlong component, chemical component of the DNA. So, nakompleto na natin si phosphate, si sugar, and then si base. So, your nitrogenous base. So, that what make up your nucleotide. Pansinin ulit sa picture sa baba, na kumpleto mo na si phosphate, si sugar, and then si base. Ang tawag niya sa kanya, kumpleto na siya, nucleotide. It is the building block of your nucleic acid. Pag pinagsama mo yung isang nucleotide, meron ka ulit nucleotide, 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 makakabuo ka ng polynucleotide. At pag pinagsama-sama mo yan, makakabuo ka ng isang linear dyan, di ba? Yan na yung component ng helix mo. Balikan natin. Kung meron tayong picture, okay, ito. Helix. May mga nucleotide dyan. Na sobrang liit lang, hindi nakita ng ating naked eye. Next, the review of the complementary base pairs. Ang sabi nga natin sa base pairs, di ba, sa helix, may mga ladder dyan. Ito yung mga nitrogenous bases natin. At dapat in base pair siya. According to Watson and Crick base pairs, dapat ang magka-partner dito, pare, si adenine tsaka si thymine. Si cytosine tsaka si guanine.
Okay, meron ako na imbentong walang kwentang mnemonic para hindi nyo makalimutan. AT, di ba? CG. At T, pahingin pera. Sige, kumuha ka dyan. Di ba? Walang kwenta. Okay, your nucleotides pa din. Nucleotides pa rin ang pag-uusapan natin dito. It is linked together by phosphodiester bonds between third prime of hydroxyl on the sugar of one nucleotide and the 5' phosphate on the sugar of another nucleotide. So, nagtataka ba kayo kung paano nagkadikit-dikit ang ating mga nucleotide dyan? So, hindi po sila basta magic lang na nagkadikit-dikit. Kasi sila it comprises of molecule pa din of an atom, of an element. So, meron pa din dapat bond na mangyari sa kanila. Okay? Balikan lang natin. It is linked together by phosphodiester bond between the third hydroxyl. So, bilugan muna natin yung una nating nucleotide. Okay? Carefully. Ito, another nucleotide. Medyo putol lang. Itong sa katabi, hindi ko nabibilugan, nucleotide pa rin yan. Okay? Between the third prime hydroxyl on the sugar, ito, bilangin natin to. Kagaya nito, gayahin natin tong sugar na to. Itong naka-blue-green sugar po yan. So, 1, 2, 3. Ito yung third prime hydroxyl. Ito daw yung binabanggit dito. And 1 nucleotide and the 5' phosphate. So, yung phosphorus phosphate group mo, di ba? Tsaka yung sugar niya, ito yung 5'. Dito naman, walang sulat, pero 5' to. So, doon daw nagko-connect yung mga nucleotide. Doon sa third hydroxyl, pansin nyo, merong linya. O, diba, nasulatan ko na hindi nyo na makita. Dito, pansinin nyo. May linya, doon sila nagkakonekta sa phosphodiester bond. Ang tawag doon, phosphodiester bond. Doon nagkonekta yung dalawang nucleotide. Kapag ka naman yung linkage between the sugar and the nitrogenous base, pansinin sa picture, ito single nucleotide lang to. Paano naman nagkadikit yung sugar mo tsaka yung base mo? Ang tawag dito, glycosidic bond. Okay? For further explanation in animation, pwede nyo i-check tong YouTube video na to. Post ko na lang sa Google Classroom. Okay? Representation pa din of our DNA, pansinin. Nakaparallel sila to one another. Yung 5' end mo dito, and then third prime end, and then third prime end dito, 5' end dito. Okay, pansinin lang, pansinin. The basic structural characteristic of your DNA as described by Mr. Watson and Crick, DNA is composed of 2 polynucleotides, anti-parallel with each other. Okay, ito sila. Pero dito, nilatag lang natin sila as linear. One strand runs from 5 prime to third prime. Okay, ito yun. And then the other strand runs from third prime to fifth prime. Okay? They are in opposite direction based on their chemical structure. Okay? On the outside, the sugar phosphate backbone. Okay, nandun siya sa may outside, di ba? Yung sugar tsaka yung phosphate natin. And then yung inyong mga hydrophobic bases, nandun sila sa loob. Nandun sila sa inner part of your DNA. Okay? Nasa outside ang ating mga sugar phosphate. Yung mga hydrophobic part ninyo, which are your nitrogenous base, nandun sa loob ng inyong DNA. The two strands are connected by hydrogen bonding between base pairs of Watson-Crick base pairs. Your adenine and thymine, 2 hydrogen bonds which are easier to separate, and then your cytosine and guanine has 3 hydrogen bonds that are harder to separate. Next, the double-stranded molecule is twisted to a form of helix with a major and minor grooves. Sabi daw nila, mas malaki daw si major groove compared kay minor groove. So, major, mas malaki kaysa kay minor. Ano naman ang significance ni major and minor groove? Based on their analysis, it is the site where the transcription factors interact. Next, the B form of DNA, first described by Watson and Crick, is right-handed and contains 10 base pairs per turn. Next, according to your scientist Erwin Chargaff, a biochemist, the Chargaff's rule is discovered by Argaff
Sherwin-Chargaff that the number of nitrogenous bases in the DNA was present in equal quantities. So, ibig sabihin, ng ratio nila is 1:1. The amount of A is equals to the amount of T according to their pair. Diba, lagi sila magka-pair. Whereas the amount of C is equals to the amount of G. Okay, parang sinulat lang niya, A is to T is equals to C is to G. Okay, in other words, the DNA of any cell from any organism should have 1:1 ratio of purine and pyrimidine bases. This means that in the laboratory, you can predict the base composition of a particular DNA but by just examining one base. Okay, mag-examine ka lang yung isang base, mape-predict mo na yung another base. Next, the adenine pairs with thymine by 2 hydrogen bonds, like what we mentioned earlier, and then the guanine pairs with cytosine by 3 hydrogen bonds. Next naman is the 3 steps performed in several cycles. So ito basically ang principle ng polymerase chain reaction. Ito yung nangyayari sa loob ng isang PCR machine. Makinig po ng maigi. Number 1 step is the denaturation of the DNA. Second step is the primer annealing. Third step, primer extension by DNA polymerase. Pakitandaan na in every step, mahalaga ang ating temperature. Mahalaga din ang mga cycle. Pakitandaan at pakitake note. In step number 1, the denaturation of your double-stranded DNA. Yung D is double-stranded to ha. The objective is to separate the double-stranded DNA into separate single strands via denaturation through the application of heat. When we increase the temperature around 95 degrees Celsius, okay, pakitandaan, heat, and then 95 degrees Celsius, the 2 DNA strands will be separated in the living cell. This is done via helicase enzyme. Hydrogen bonds between the 2 strands will be broken. Okay? Kasi po, merong mga hydrogen bonds sa ating DNA. Magra-rupture siya once na in-increase mo yung temperature. So, ang tendency, maghihiwalay sila, magsiseparate yung strand nila. Ano bang purpose? Ano ba yung next step natin? Bakit kailangan mong paghiwalayin yung strand ng ating DNA? Let's find out. On your step number 2, the primer annealing. The synthetic primers performed and are not produced by primase binds to the regions that you wish to amplify. Pakitandaan yung keyword amplify. The temperature is lower about 40 degrees to 60 degrees Celsius. So from 95 degrees, ilo-lower na natin yung temperature sa annealing natin. Ibababaan na natin siya sa 40 to 60 degrees Celsius. And then on your step number 3, the primer extension by DNA polymerase, your primers will be extended by DNA polymerase synthesizing your desired DNA sequence. It is carried out at 72 degrees Celsius. Pakitandaan, the DNA polymerase from Thermus aquaticus, it is your top DNA polymerase, is used because it can withstand high temperature. So ano po ba yung sinasabi niyo, ma'am? Hindi namin ma-envision. Ito siya. So sa loob ng inyong polymerase chain machine, reaction machine, ang step number 1 mo is i-denature natin siya through the application of heat. So pag naghiwalay na yung mga strand mo, ang step number 2 nun is mag-go through na siya sa annealing. Ang temperature natin dyan is 40 to 60 degrees, which in this case is 55 degrees. Okay? Susunod nun, in preparation na siya sa amplification. Okay? Kakabit na yung mga Taq polymerase niya dyan. Kasi, kaya Taq polymerase yung napili na ating polymerase dyan kasi it can withstand high temperature. Pansinin nyo itong mga temperature natin inside the polymerase chain reaction machine, matataas talaga yung temperature niya. Okay? Kasi nga the goal is to separate the 2 strands of your DNA. Once na nakakabit na yung Taq polymerase niya, it will be subjected to amplification. Ano ba yung ibig sabihin ng amplification? Pansinin po, meron kang 2 strand dyan. Mag-a-amplify na yan, dadami na yan. Kunwari, nag-produce yan ng 2. Yung 2 yan, magpo-produce na naman ng 3. Yung 3, magpo-produce naman na ng 4. Okay, hanggang dumami na siya. And then, it will be repeated in the cycle of 20 to 40 times. Pakitandaan yung mga cycle, pakitandaan yung temperature. There are usually 30 cycles in performing your PCR, taking about 4 hours. Okay? Kasi nga, ang goal ng ating polymerase chain reaction at the end of the cycle is to make 1 billion copies of your DNA strands. Pansinin ang RT-PCR. RT-PCR. It can be read as real-time PCR or reverse transcriptase. Reverse transcriptase naman kasi applicable kasi ang ating mga coronaviruses, yung mga SARS-CoV-2 natin, RNA virus sila. Katulad sa central dogma, from RNA, i-convert mo sila to DNA and then mag-go through dun sa process ng denaturation, annealing, extension. RT, pwede ding real-time kasi within 4 hours, nababasa na yung graph doon sa amplification. Kunwari, meron kang graph. Pag nag-amplify tumaas, ibig sabihin readable as positive. Parang ganon. Discuss na lang natin siya much further on next discussion or next topics. Okay, yung RT-PCR mo, pwedeng reverse transcriptase, yes. Pwede ding real-time kasi within 4 hours, nakikita mo na yung pag-amplify. Ibig sabihin, nung pinadami mo yung DNA, meron talagang presence ng DNA ng SARS-CoV-2, for example. Yun yung principle ng mechanism ng ating polymerase chain reaction. And that concludes our discussion for today. These are my references. Thank you and God bless.